So, dear scholars, previously we have been handling the dihybrid cross, and it is what we are still moving with. So now, the other time we dealt with the F1 generation of the dihybrid cross, where we crossed a tall red flowered plant with a short white flowered plant. And we observed that all the offspring in the F1 generation were tall and red flowered, an indicator that the allele for tall character was dominant to that for short character. And then the allele for red character was also dominant to that for white character. So now, having known that, we are going to proceed with the F2 generation. And so this is the F2 generation as indicated here. In F2 generation, second filial generation, we are selfing the F1 offspring, that is F1 crossed with F1. Of course, briefly here, this is the same as self-pollinating two F1 individuals as shown on the board. So, parent phenotype will have tall red flowered plant. It is being crossed with tall red flowered plant. Then, parental genotype, which are, of course, the parents are diploid. The genotypes are here. We have this genotype here. They are heterozygotes, as shown on the board. Actually, both parents are heterozygotes. And now, having written down the genes, as shown on the board, what follows is separation of the genes, also called segregation of the genes, followed by independent assortment, rearrangement of the genes to form new allele combinations, as shown here. And of course, briefly I have been saying that if you want to form them separately, this is how you can do it. Genes segregate and then recombine or assort independently. So this allele, dominant allele T, can combine with this one here to form this allele combination, and also the same gene T can combine with small r to form capital T, small r. This one will give us T r as shown on the board, like that, such that the gametes or the possible allele combination will be T, capital R, then capital T, small r, small t, capital R, and small t, small r. Those are the gametes, or those alleles, those combinations of the alleles are carried in gametes, which are haploid, denoted by N. The segregation and independent assortment occurs during meiosis. Meiosis occurs like that. We shall have the same allele combinations even for the second parent, since they have the same genotype. So we shall have capital T, capital R. Then we shall also have a capital T, small r, and then small t, capital R, and small t, small r. Those are the possible allele combinations. So now, What follows next is fusion of the gametes. The gametes fuse together, and the fusion is random. There is no order. So we say that each member of an allele pair can randomly combine with the allele of another gene pair. So now what will happen here is that since the gametes are many, what we are going to do is that we are going to use a Punnet square. We shall use a Punnet square to carry out our crosses. Punnet square. A Punnet square is a grid named after a British geneticist. This man was called R.C. Punnet. It was named after a British geneticist called R.C. Punnet, and he called it a Punnet square. A Punnet square is a grid with vertical squares and then horizontal squares. That's what we call a Punnet square. R.C. Punnet, of course, was based at Cambridge University. That is where he did that work. So now, random fertilization. Random fertilization. Here I'm saying we are going to use a Punnet square. A Punnet square minimizes errors that can be made when we are crossing many gametes. So we use a Punnet square to minimize all the errors. So I am going to draw the Punnet square on the board as shown here. Punnet square. I am going to draw a Punnet square. So if I can draw it like that, and then I have said a Punnet square consists of vertical squares and then horizontal squares. So if I can draw my vertical squares, shown here on the board, vertical squares. Now, in the vertical squares, we first possibly complete it. In the vertical squares, we normally write there the male gametes, and the male gametes are represented using this symbol. Wherever you see that in genetic studies, it means male gametes. Then this one here, this symbol here, means female gametes. So the female gametes are normally written down within the horizontal squares. So now, let me assume that one of these parents is male and the other one is female. So I'll write the male gametes here. We have TR, then this one is capital T also, TR, then small t, capital R, and then small t, small r. Then female gametes, we shall have the same. We shall have capital T, capital R. Then we shall also have capital T, small r. I'll also have small t, capital R, and then lastly small t, small r. Those are the female gametes. And then I seal my square. I seal my square. So I have so far drawn what we call the vertical squares. Let me also put the horizontal squares. Horizontal squares as shown on the board. So of course when we are crossing many gametes, then we simplify the crossing by use of a Punnet square. So now we can cross having looked at the Punnet square as shown here. Now, when this gamete fuses with this one here, what will happen? We shall form an offspring with a genotype. With the genotype capital T, then capital R, capital R. This one here will be capital T, capital T, then capital R and small r. This one will be capital T, then small t, capital T, small t, and shall have capital T, small t, capital R, capital R. This one will give us capital T, small t, and then capital R, small r, like that. These gametes, when this gamete fuses with this one here, we shall form capital T, then capital R, small r. This one will give us capital T, small r, capital T, small r. This offspring will bear a genotype of capital T, small t, then capital R, small r. This one will have genotype of capital T, small t, capital T, small t, then small r, small r, small r, small r. That is it. Then here we shall have an offspring with genotype capital T, small t, capital R. This one here will give us capital T, small t, then capital R, small r. This offspring will bear genotype of small t, then capital R, capital. This one will bear small t, then capital R, and then small r. Then lastly, this offspring will bear a genotype of capital T, small t, then capital R, small r. This one will bear capital T, small t, then small r. This one will bear a genotype of small t, then capital R, small r. This one here will bear a genotype of small t, then small r. Having done that, the other important thing is to ensure that you give an identifiable mark to the offspring bearing the same genotype, so that the mark can help you to identify the possible genotypes of the offspring in the Punnett square. So other than writing full words there, we can give them specific marks, which marks will then help us to identify the possible phenotypes of the offspring. Now, this one is tall. It is tall and red. There it is tall and bears red flowers. If I can give it that mark as shown on the board, any plant or offspring, any plant or individual which will be tall and bears red flowers, I'm giving it that identifiable mark. The same applies to this one here. It's also tall and will bear red flowers. The same applies to this one here. It will be given the same mark shown on the board. Then also this one here will be given the same mark because it is also tall and will bear white and will bear red flowers. The same applies to this one here, tall, and once it matures, then it can bear red flowers. Then also this one here, it will be tall and can bear red flowers. This offspring is also tall and can bear red flowers. The same applies to this one.